Welcome to Broad Eye, the podcast that explores knowledge gaps in ophthalmology and eye care. Hello, everyone, and welcome to another episode of the Broad Eye podcast. My name is Sean Maloney, and today I am sitting with Dr. Mark Pennesi. Dr. Pennesi is a professor of ophthalmology at the KCI Institute of the Oregon Health and Science University. Dr. Pennesi, welcome to the show.
Thank you, Sean. It's a pleasure to be here today.
Thank you. So we're going to dive into a topic that is certainly caught a lot of headlines in recent years. We're talking about technology called CRISPR, and specifically how CRISPR is being used to treat rare eye diseases. But I was hoping maybe you could start off with just a high-level overview of what CRISPR is for people who are listening.
Sure. Well, I think it's probably useful to start off with a high-level overview of genetic diseases and how those occur, and many will be familiar with those concepts, but there are many different genetic diseases, and of course these result when you have a mutation in the DNA. DNA codes for RNA, which codes for proteins, which makes up all of the cells in our body. And so there are different kinds of mutations. You can have mutations where you essentially lose function of a protein, or a protein doesn't get expressed. You can also have gain-of-function mutations where the protein takes on properties that are toxic to the cell or cause problems with the cell. So when we think about gene therapy, there are different kinds of gene therapy. So traditionally, we've talked about gene replacement or gene augmentation therapy, where essentially you're putting in a new copy of the gene. Usually the way we do that is delivering a copy of the gene using a viral vector, and a small piece of DNA will go into the nucleus and code for a normal copy of the protein, thus augmenting the missing protein or replacing what is defective. With gene editing, it's a little different, because instead of putting in a new copy of the gene, we're actually editing the native DNA or fixing the problem where it occurs. And there are different ways to do that and many different mechanisms to do that. I think in the simplest manner, you can make a cut in the DNA and take out a piece that is causing problems. And so CRISPR is a technology, and this is something that essentially comes from bacteria. There are proteins that bacteria have called Cas proteins that are used to cut up pieces of viral DNA that try to infect the bacterial cells. And it's really a primitive immune system for bacterial cells to protect themselves from viruses. And they actually can keep pieces of viral DNA in their own DNA, almost like a memory bank, so that in the future they can recognize those viral pieces of DNA and cut them up more efficiently. So what can be done then is sort of co-opt that system for use in the human genome. So we can re-engineer the Cas proteins and target them to go to a spot where a mutation is, and make a little cut in the DNA, a double-stranded cut. And if you have two different guide RNAs, you can actually cut out a piece of DNA. So in a situation where, let's say, for example, you might have a stop codon or a mutation that is creating a stop codon, you might be able to cut that out, and that can result in full-length transcription of the protein. So that's kind of a big overview. There are many different variations on that, and more advanced ways of editing the DNA that are also being looked at.
No, so thanks for that's a nice overview. And what I was hoping we're going to talk about a couple of specific diseases, but before we do that, you alluded to the fact that there are different ways of approaching gene therapy, and gene editing is one of those ways. And certainly, a new and exciting way. What are some of the benefits, I guess you could say, or cases where gene editing is maybe the method of choice to pursue clinically? In what diseases or in what situations is that a better option than maybe traditional gene therapy?
Sure. Well, I think, there are two situations that come to mind. First, when we think about gene augmentation therapy, where we're putting in a new copy of the gene, typically that DNA forms an episome or a ring inside of the nucleus where it will then drive expression of the protein. And well, we think that in theory that DNA could express forever. In reality, we don't really know. And certainly it's possible that over time that replaced gene or augmented gene could be silenced and you could lose expression. So one advantage of actually editing the native DNA would be that you would get longer persistence. The second issue, I think, becomes one of regulatory control. When we put in a gene for gene augmentation, we're putting a promoter usually to drive expression. And in some cases, you can use the native promoter, but in other cases, you might have to use a constitutive promoter that drives strong expression. But you're losing the context of other elements in the DNA that might regulate the expression. So with gene editing, though, because you're making the change at the native location, you're sort of maintaining all of those regulatory elements. So you might actually get a more accurate gene expression as compared to gene augmentation. And that might be important in a situation where overexpression of the gene might be toxic. So, if we look at, certain diseases, for example, with RP65 related retinopathy, that wasn't an issue because in patients who have Leber Congenital Amaurosis due to RP65 mutations, you can overexpress RP65 and it's not toxic to the cell. But we know that other proteins, that can be a problem. And so in a situation where you're driving expression and you don't have as much control, that could actually create issues. And so gene editing might be better in those situations.
Okay, no, that makes sense. Now, is that just as my own curiosity? I'm going to go all over here as you're giving answers, but is that something that is still being heavily researched, like in these various degenerative retinal conditions, and within the same condition, there could be various underlying genetic issues? Is this something that's still being heavily researched to understand? Yeah, if like overexpression of this is, because imagine for some of them it's probably a sweet spot, and like it gets to a certain point, then it's too much of a good thing is a bad thing in that scenario, right?
Absolutely, and I think that it's not the easiest thing to study because you can certainly look at it in mouse models by trying to make different lines of mice that have different expression levels, or there are ways, of course, to have inducible promoters that will drive different levels of expression. But that's a lot of work and a lot of investment of time. So we have knowledge of that for certain genes, but when we think about inherited retinal dystrophies, there are probably over 300 different genes that are involved, and we certainly don't have that information for every gene.
Oh, that's fair. We're going to talk a little bit about Leber Congenital Amaurosis, or LCA. I was hoping you can give a little overview of what LCA is, and why it is an attractive, I guess, starting point for gene editing in the eye.
Sure. So Leber Congenital Amaurosis is really a collection of diseases because there are probably about 25 different genes that can cause that particular phenotype. And we really define it as a very early and severe retinal degeneration. Oftentimes, patients will present within the first year of life with severe vision loss, nystagmus, photophobia in some cases, or problems in the dark. It can also present in a less severe form in the first 5 years of life, but it typically is affecting infants and young children. Additionally, it's a progressive disease. So they start off quite severe and it gets worse with time. And so, the majority of these patients will ultimately be legally blind at a young age. And some will even lose all vision and go completely blind. So it's really a devastating disease in that sense. I think the reason that it's been a target for gene therapy and gene editing is because of the potential to really restore vision at an early age or to maintain vision. And if you think about it, a lifetime of blindness is really a burden, incredible burden on the patients, but also on society. The economic impact is very large. So the other reason that it's a good disease to look at is there are certain genes that cause LCA that have what we call structure function dissociation. And what that means is that if we look at the retina of these patients, we can see that there's still cells there, but they're not working. So these patients may have very poor vision, light perception vision, but you look in the center of the retina and you can see that there are still photoreceptors there, but they don't have outer segments, so they can't actually capture light. And this often happens with certain proteins that are involved in the cilia. And that's really kind of the ideal situation for a therapy, because then not only might you have the ability to slow down degeneration, but you might actually have the ability to restore vision. If you can replace that protein that is important for elaborating and elongating the outer segment, the regrowth of the outer segment could actually bring back vision.
Thanks for that explanation. I gotta imagine that when you're mentioning that it's not an obvious, I guess, but an attractive choice because you can potentially give people back vision, and which can be early on, that can affect them their whole life. Gotta imagine the other side of that coin, though, is that these procedures are probably happening mostly in kids. Is that right?
So, we actually always start in adults for safety reasons. And so many of the initial studies during the dose escalation are performed in adults. And there's still hope with those patients that they might actually gain vision back. We, of course, think that the potential is greater in children for a variety of reasons that I can get into. But there is still actually benefit, and we've seen that in many cases, in particular with Beretagene and Naparvovec. We've seen lots of benefit in adults, and even from the preliminary data from the BRILLIANCE trial, we've actually seen improvement in some of the adult patients. So I think that's very exciting, and we're hopeful that as we go to younger and younger patients, that we're going to see an even greater benefit.
No, that's great. And you alluded to that trial, so I thought maybe we could dive into that a little bit. If you could just give a little context of what the trial's all about, what results that we have that you can share publicly now, and then maybe where it's headed.
Mm-hmm. Yeah, so the BRILLIANCE trial is looking at patients that have a particular form of Leber Congenital Amaurosis, or LCA type 10. And this is patients who have mutations in a gene called CEP290 or CEP290. And in particular, it's looking at a specific mutation, which is an intronic mutation that affects splicing. So the goal in this trial is to treat those patients by using gene editing to essentially cut out the portion of the RNA that kind of acts like a little stop sign. So there's a mutation that essentially creates a stop sign so that you don't get full-length transcription, and you result in a protein that's too short and probably gets degraded. But if you can use gene editing to cut that out, you can actually induce the cell to make the full-length protein. And so we've treated a number of patients. Some of that data has been presented in the first 7 patients, and I think it's actually very exciting. It's still preliminary, of course. This is a phase 1/2 trial starting off in very severe patients, first at a very low dose and then moving up to a medium and higher dose. But even one of the patients who was in the low dose actually had subjective improvement in their vision. And one of the patients in the medium doses that we treated at our site actually had an improvement in their visual acuity. They were able to detect light easier when we tested their sensitivity. And even on a mobility test where we have them essentially navigate through a kind of a real life, maze, they were able to perform that better after treatment. And so, very early on, we're starting to see signs of benefit, and that's very exciting because the hope would be that as we go to higher doses, we might get better effect. And then additionally, as we go to younger patients, we might see added effect.
No, so I find it, super interesting because gene editing is obviously a hot topic, if not the hot topic in medicine right now. And it's not just for, inherited retinal diseases, right? So I guess, why is the eye, this is the first time, my understanding, that this is actually being done in humans. I mean, cells before were being taken into a dish, essentially into a petri dish, where this gene editing was taking place and then put back into patients. But this is the first time, to my knowledge, that this has ever been done directly in patients. So maybe correct me if I'm wrong on that. And if you could just
the first really in vivo treatment. So you certainly can take blood cells out and do gene editing in the dish and then put those cells back in. And, that's a great way, to treat, certain bloodborne diseases. There are definitely advantages to that method, but for something like the retina, we can't take the retina out of the body and put it back in. And the same goes for the central nervous system. So we do need to have ways of treating organs that are not removable. So the eye has a lot of advantages when it comes to gene therapy. First off, we have two eyes, so we can treat one eye in an early phase trial and compare it to the untreated eye. The second is that the retina is essentially part of the brain. It's an extension of the brain. So we really are treating the central nervous system, but we can look in and actually see the retina, unlike other parts of the nervous system. And we have the ability then to do a lot of imaging where we can actually see the histology almost on a cellular level using a technology called optical coherence tomography. And that gives us an advantage to actually see if there's any inflammation, almost in real time. And that's a final advantage of the eye, is that because the retina is behind the blood-brain barrier or the blood-retinal barrier, there's a relative immunoprivilege in the subretinal space, which helps prevent inflammation towards the viral vectors that we use to deliver the gene editing machinery. So those are some of the advantages of doing this in the eye.
No, I think, and it's it's super exciting as the whole medical community looks on to see what happens in the eye, because this seems to be an initial entrance point for for in vivo therapy, and for the reasons you've outlined. And I think it's just exciting. The early results are super exciting, beyond well beyond the ophthalmology community. This technique, is this something that can be used in other forms of inherited retinal disease? And if so, which ones do you think would be most amenable to it?
Oh, absolutely. This is a technology that's going to be able to be adapted to other genetic mutations. I think that the power in this technology really is applicable where the genes are very large and they are not really amenable to gene replacement therapy. So, as you know, there has been, one drug approved for gene replacement, and that's a very small gene, RP65. And so that gene can fit within a AAV vector. But there are common genes such as USH2A that cause retinitis pigmentosa that are really too large to fit into an AAV. And so in that case, gene editing is going to probably be a better approach.
Yeah, no, I think it makes a lot of sense, because I think people have tried to tackle that problem in other ways by, trying to have the cells express truncated proteins, or looking at other, lentiviral vector systems and whatnot. It seems like this gene editing approach seems to be a potentially a much more robust solution to the problem of trying to package large genes and deliver them into the cells. So I think it makes a lot of sense. Now, is this something that your group is uniquely spearheading when it comes to LCA and the eye, or are there other interests and groups doing this as well?
Well, I certainly don't want to take the credit for this. I think that this is really represents years of work by the scientists who really developed, this technology, such as Jennifer Doudna and, others who, have really been recognized for that, as well as, my collaborators on the trial, Eric Pierce at Mass Eye and Ear. So we're all working together. I mean, this is really a big group, effort of many different people. And, there are other people looking at, gene editing for treatment of other genes. So I think that this is certainly, something that is going to really continue to grow and evolve. And I hope so, because I think there is just such potential for helping patients.
And that's great. I just want to maybe wrap up with one more question, if you don't mind. And it's more just your thoughts on where this is all headed, and maybe, I guess, on the BRILLIANCE trial, what are some of the next steps? But then, from your vantage point, where is this all headed? And I won't ask you to speculate on timelines, because I think people always hate to speculate on timelines to the clinic. But I guess is the research on track with where you and your team all hoped it would be now? And are the next steps in view?
I think we're definitely, on track, and I think things are really, moving forward, especially when you factor in, we've been in a 2-year pandemic, which hasn't been the easiest thing for clinical trials. But I'm happy to report that things are moving ahead. And, as I mentioned, we're really excited to hopefully be treating younger and younger, patients as time goes on.
Excellent. Well, listen, I want to thank you for, first of all, for, helping to spearhead this work. I know you're passing the credit to a lot of other people, but certainly you're central in this. So thank you for doing that. As from my perspective as a patient, I appreciate that. But I also like to thank you for coming on the podcast today, sharing your thoughts and perspectives. It's certainly something that I enjoyed, and I think that the audience is going to enjoy as well. So thank you.
Thank you. It's been a pleasure.
And that concludes today's episode of the Broad Eye Podcast. If you enjoyed this episode, please be sure to subscribe on your favorite podcast platform. Of course, ratings and reviews are always welcome. And you can certainly share this episode with any of your colleagues or friends who might enjoy it. Thanks for listening.